Hello, my name is Ryan Thompson, and today I will be presenting on the Trypanosoma brucei species of parasite. On the left hand side of the first slide, you can see a close-up photo of a tsetse fly, the primary vector of Trypanosoma brucei species. And on the right, you can see a scanning electron micrograph of a Trypanosoma individual contained within a blood smear. Now, Trypanosoma brucei is the primary cause of African sleeping sickness, which, if left untreated, is a 100% fatality rate. And in that way, it is similar to that of rabies in terms of deadliness. It's a eukaryotic organism. It's microscopic, and it primarily likes to survive in warm, humid areas of sub-Saharan Africa. There are two different forms of this disease: rapid onset human African trypanosomiasis, or HAT, and chronic human African trypanosomiasis, which are both caused by two different subspecies of the disease, which I'll go over later. Several epidemics have occurred in the past century, resulting in the deaths of several 100,000 people due to the location of these epidemics and the time from when these happened in the early 1900s. It's difficult to gain exact data on how many people died, but suffice to say that it is a very serious disease. Now, due to its dependence on the tsetse fly for being its mechanism of infection, it has a range that resembles the range of the tsetse fly. There are two subspecies: Trypanosoma brucei rhodesiens, which primarily is located on the east coast of Central Africa, and then Trypanosoma brucei gambiense, which is primarily located in the Democratic Republic of the Congo, and it is a lot larger range. Trypanosoma brucei rhodesiens is going to cause rapid onset HAT, and then Trypanosoma brucei gambiense will cause chronic HAT. Now, for the taxonomy of Trypanosoma brucei, it's an organism of the domain Eukaryota, of the phylum Euglenozoa, of the class Kinetoplastea, of the order Trypanosomatida, of the family Trypanosomatidae, of the genus Trypanosoma, and of the species Brucei, the two subspecies gambiense and rhodesiense. The life cycle of this parasite begins when a tsetse fly bites a reservoir, such as a human or cattle or ungulates, in the case of the rhodesiense subspecies. It's injected into the bloodstream in the metacyclic trypamastigote form, and it replicates via binary fission until a blood meal is taken again by its tsetse fly. In the midgut, it's going to develop through its procyclic form, its epimastigote form, and back into its metacyclic form, at which point it is going to become infective once again and can be transmitted through another bite of a tsetse fly. When it is shown under a microscope, it's roughly the size of two red blood cells, and on the right side of the slide, you can see that there is a scanning electron micrograph of one of these individuals, and it has two very long folds that the parasite uses to propel itself through whatever surface it is encountering, and to transport itself to areas of greater nutrition and out of danger. There are several factors that determine the virulence of this parasite. The first and primary one that I will be discussing is the avoidance of antibody-dependent complement-mediated destruction, in which it can vary the antigens it presents on its cell membrane, dodging the antibody-dependent recognition by the immune system. The second and third are primarily involving the secretion of exoproteins from the Trypanosoma brucei, which can prevent the activation of your helper T cells and interfere with B cell function. As you can see in the diagram here, there is a certain genetic makeup of protein that is being expressed on the cell membrane of the Trypanosoma species in the top left corner. And then over time, this antigen is going to be replaced with a randomly genetically varied copy of this VSG. Now, VSG stands for variable surface glycoproteins. It is a long chain of sugars mixed with protein that is going to be coating the outside of the organism. And the genes that code for this can be extremely rapidly changed and varied, and the organism cycles through a lot of different variations in order to avoid detection by the host immune system. 2,000 genes control this process, and once you factor in splicing of the mRNA, there's functionally limitless variations of glycoproteins that can be expressed. An added benefit is that the glycoproteins can prevent the adsorption of complement and other antibodies that might be able to recognize those more invariable parts of the cell membrane, like aquaglycoproteins. If I was going to explain this to a patient, I would say that the infection is acquired from the bite of a tsetse fly, which is native to Central Africa. It's a microscopic worm that replicates in the blood and can spread throughout the rest of the body. It avoids detection by the immune system by coating itself in a shifting coat made up of proteins and sugars that prevent it from being recognized by the immune system, and it can secrete a bunch of proteins that can interfere with how the immune system works even more than it already does. And this spreads from person to person via the bite of a tsetse fly when a fly bites an infected individual. Now, there are several treatment options. The primary and the most recommended is fexinidazole, which is reduced by the parasite's nitroreductases into two highly reactive metabolic forms called sulfoxide and sulfone, which I'll discuss later. But basically, these both react with the parasite's DNA and proteins. The second is suramin, which inhibits the enzymes that are responsible for oxidizing NADPH, which interferes with critical cellular functions. And the third is NECT, which is an acronym for nifurtimox eflornithine combination therapy. Nifurtimox is a nitroheterocyclic trypanoside, and it produces superoxide radicals, which damages pretty much everything. And then eflornithine is a permanent inhibitor of ornithine decarboxylase, and that essentially prevents the trypanosome from replicating any further. Now, the mechanism of action of fexinidazole is really interesting. It's an orally administered drug, and it is not effective until it is processed by the trypanosome. It treats both stage 1 and stage 2 of infection because it has the ability to cross the blood-brain barrier. Now, the trypanosome produces a nitroreductase, which, when it reduces the fexinidazole, it produces two metabolites known as sulfoxide and sulfone. Both of these are reactive enough to significantly damage the electron transport chain of the trypanosome's mitochondria, and also do significant damage to the kDNA, which is the way that trypanosomes store their genetic information. Both of these will sufficiently disrupt the metabolic processes within the parasite in order to destroy it. Now, we have discovered some strains of this parasite that do have drug resistances. The two of which that I will be discussing today is larsuprol and pentamidine. I did not discuss these drugs in detail because they have extremely high side effects, and also we are finding species that are resistant to them anyway. But the mechanism of resistance for this is a surface protein that is going to be it's functions a lot like an efflux pump for bacteria. In this case, it is an MRPA ABC transporter transporting larsuprol and pentamidine out of the cell. So as you can see in this image here, pentamidine and larsuprol will both be admitted entry into the cell by the surface transporters P2 or AQPT, and before it has the chance to administer its effect, it is pumped out of the cell by the MRPA protein. All right, here's my works cited. Thank you very much.